I'm Dr. Richard Visconti, and I know stem cells. So my laboratory is focused on the use of stem cells for cardiovascular regenerative medicine. And two of the major projects in the lab deal with myocardial infarction repair and also generation or bioprinting of blood vessel segments for bypass surgeries or vascularization of bioengineered organs. So in terms of myocardial infarction, when one has a heart attack, that area of the heart suffers death of the muscle and the blood vessels in that area that help the heart perform its function, which is to pump blood to all of the organs and tissues in the body. This is absolutely necessary because blood carries all of the food and nutrients that cells need and removes waste that can be eliminated from the body. So what our hope is with stem cells is that we can use the talent that stem cells have, their ability to differentiate into desired cell types, to actually repair that dead portion of the heart that is produced by a heart attack. So we use bone marrow-derived stem cells to differentiate into cardiac muscle cells, vascular cells, and even cardiac fibroblasts, which support the cardiac muscle. And our hope is that one day we can either generate replacement tissue or stimulate stem cells that reside within a patient's body to go to the area that's been damaged by the heart attack and actually turn over or replace the dead, damaged tissue with healthy, functional tissue, which will result in a better quality and quantity of life for the patient. In terms of printing vascular structures, we are interested in taking advantage of the ability of stem cells to differentiate into the cells that make up blood vessels so that we can bioprint blood vessels of the desired diameter and length so that they can be used in the clinic for bypass surgeries, for example, when a patient has atherosclerosis or other coronary blockage or vessel blockage disease, we can use that patient's stem cells to generate blood vessels to bypass their diseased or damaged blood vessels. Our ultimate goal is to one day be able to bioprint a vascular tree, so that would be a hierarchically branched system of blood vessels from very large to very small in diameter, small as a capillary in fact, so that we can use these bioprinted vascular trees to serve as the substrate or scaffold onto which we can print organ-specific cells like liver cells, heart cells, bladder cells, even intestinal cells, so that we have a vascularized, bioengineered organ at the end of this process that can be implanted into a patient who needs this replacement organ. And the added advantage here is if we use postnatal stem cells that are harvested from that patient's body will not reject the organ because it's been made from their own cells. So patients will have the benefit of restored function of that tissue or organ without the need to be on immunosuppressive therapy. That means they won't have to take drugs for the rest of their life that will prevent their body from rejecting that organ because that organ will in fact be made from their cells.